What we're going to do today is I'll finish the story that we started last time on the parietal cells, and I think I pretty well got through it in this section, but I didn't in the first one. So I'll review the idea of the G-protein coupled receptors and how those G-protein coupled receptors are impinging in parallel pathways onto the generation of acid. And we'll talk a little bit about the idea that there are the three major G-protein coupled receptors, one for acetylcholine, which is released from postganglionic parasympathetic neurons, one for histamine released from the enterochromaffin-like cells, and a third one that binds to gastrin released by G cells. And we'll try to bring that all together and talk a little bit about how those G-protein coupled receptors in the parietal cells are governing the release of hydrogen ion. And I'll just give you a few words here on the clinical implications for this, that we have major forms of antacids that we take all the time. You go to the grocery store and buy these actually. One is cimetidine. And it's instructive because cimetidine is actually an H2 receptor blocker. What it's doing is it's preventing the action of the histamine released from the ECL cells simply by binding to the site that's normally bound to by the histamine, and the histamine then is no longer able to bind to that G-protein coupled receptors. That's cimetidine. The more modern form of an antacid is something called omeprazole. But it's instructive to think about the way it works because what the omeprazole does, and these are prilosec and Nexium, these molecules bind directly to the pump itself. And when these molecules bind to the pump, they inactivate the pump. They basically turn it off. And they molecularly turn it off in such a way that it has to be synthesized again to become active. So you can't reactivate that pump. It's a one-way trip. So what that tells us from biology is the omeprazole is going to take the pump out of the circuit, and then we have to upregulate genes in order to make more protein so we can put those pumps into the apical membranes of the parietal cells to generate acid. And we'll talk a little bit about that. We want to spend most of our time, though, on the small intestine because, after all, that's the next stop along the GI tract for the fluids that's in the stomach. We said yesterday that we had chyme in the lumen of the stomach. The chyme commonly had a pH down around 2. And we were going to then, what we'll see today is we're going to squirt a bolus of that fluid across the pyloric sphincter into the small intestine. Under normal circumstances, the pyloric sphincter, like the esophageal sphincter, remains tightly closed. And then there's a control, there's a signal from the brainstem that says open up, and it opens up and allows about 30 to 50 mils of chyme into the small intestine. But a couple of things have to happen pretty quickly in the small intestine. First is that all of the enzymes we're going to see that are digestive enzymes in the small intestine have a pH optimum of around 7.2 to 7.4. So the contents of the stomach at pH 2 basically will destroy those enzymes. So one of the things we have to do is we have to bring the pH from about 2 up to about 7.2 to 7.4. That's the role of bicarbonate that's going to be released from a series of cells in the pancreas called duct cells. Similarly, the contents of the stomach will be enriched for lipids and fat and starch, and we're then going to begin digesting those, and we're going to see there are a set of cells in the pancreas called the acinar cells that will release digestive enzymes. But before we get to the interplay between the small intestine and the pancreas, we have to spend a little bit of time just on some crude anatomy. And I'll walk through the structure of the small intestine in terms of the tunics. And we'll see that the tunics, or layers of the small intestine, are reminiscent of what we talked about yesterday with the stomach. You have the same kind of arrangement of mucosa and submucosa and muscle layers and then the serosa or mesothelium. But the intestine is a pretty long structure, and so it's broken down into three major regions, the duodenum, the jejunum, and the ileum. And so we'll just draw this out. And then finally, where we want to end up is a description of the villi. The villi are these finger-like projections, but they're multicellular projections. The villi are composed of hundreds of thousands, perhaps millions, of individual epithelial cells called enterocytes. And these finger-like projections are hollow. And they're organized in such a way that the epithelial cells, the enterocytes of the villi, have their apical surfaces facing the lumen of the small intestine, and the basilar sides are facing a series of capillaries that are branches of the hepatic portal system. And we'll talk a little bit about that. So again, hold on to the idea that the villus is a multicellular structure composed of thousands and thousands of enterocytes, and that the enterocytes are oriented in such a way that the apical surfaces are facing the lumen of the tube, but the basolateral surfaces are facing a series of blood vessels. They're basically facing the endothelial cells of blood vessels, which are branches of the hepatic portal vein. So we have a tremendous blood supply flowing through the gut. And this blood flowing through the gut is there primarily to pick up nutrients that have crossed the epithelium, crossed the mucosa of the small intestine. Those nutrients will go into the hepatic portal system and be off to the liver. One of the ideas we'll try to bring up today and hopefully finish on Monday is that when we're talking about the blood vessels, when we're talking about the hepatic portal system, we're talking about the movement of water-soluble molecules. We're talking about the movement of amino acids and glucose. Lipids follow a whole different pathway. We're going to find that in the enterocytes, lipids are actually broken down and reconstructed. Those lipids are then released from the basolateral surface as something called chylomicrons. And the chylomicrons are enormous. They're like a micron in diameter. They're so big that they can't pass between the narrow gaps that exist between endothelial cells of the hepatic portal system. So instead, what we do is we'll pass these lipids as chylomicrons into lymphatic capillaries. And then that lymph will go back into the blood, and we'll talk a little bit about that. But before we get to the absorption part across the epithelium, the enterocytes, we need to talk a little bit about the hormonal control, the interplay between the small intestine and the pancreas. And what I want to emphasize here are just a handful of cell types. There are S cells and I cells in the intestinal mucosa. S cells release a hormone called secretin. I cells release a hormone called cholecystokinin. The target for the secretin and the cholecystokinin are epithelial cells in the pancreas. The secretin released by S cells will bind to pancreatic duct cells and end up releasing bicarbonate. The bicarbonate released from the epithelium of the pancreatic duct cells, or the bicarbonate released from the apical membranes of the pancreatic duct cells is now destined to go right into the lumen of the small intestine. That bicarbonate now is going to serve as a buffer. It's going to serve as a buffer for the hydrogen ion that was put into the stomach contents by the parietal cells. And as we walk through this, one of the things you'll recognize is that these pancreatic duct cells are basically parietal cells turned upside down. Where the parietal cells are releasing hydrogen ion from their apical membranes, these pancreatic duct cells are now releasing bicarbonate from their apical membranes and releasing hydrogen ion from their basilar membranes. And we'll use that bicarbonate then to neutralize the acid in the small intestine. Then there are another set of cells, the pancreatic acinar cells, and the function of the pancreatic acinar cells is now to release digestive enzymes. And we'll see that there's a signal between the epithelium, the mucosa of the small intestine, and the acinar cells through cholecystokinin released by the I cells. And we'll talk about that. And finally, if there's time, we'll talk about digestion in the intestine, and we're going to break it into bulk phase in the sort of center of the tube and the brush border, which is right along the edge. But let's start here and sort of try to finish up where we were yesterday. I'm going to draw this cell as a parietal cell. This is, of course, in the stomach mucosa. And to remember that we spent a lot of time yesterday describing the existence of these blind alleys, and that in the transmembrane proteins in the membrane of these blind alleys was a hydrogen ion potassium ATPase. And the hydrogen ion potassium ATPase worked in concert with a series of potassium channels. And the physiology of this arrangement is we allowed for the potassium to flow down its concentration gradient into these blind alleys. Then the potassium would bind to the extracellular side of the transporter, where in the cytoplasmic side, we bound a hydrogen ion. And because this was an antiporter, the hydrogen ion was kicked out and the potassium came in. And we closed with the idea that there were three major G protein coupled receptors that were regulating this event. The first one, I'll put it in orange, was a GPCR that bound or binds gastrin. Gastrin is a peptide hormone. Gastrin is a peptide hormone released from the G cells. And these were nearby cells to these parietal cells in the gastric pits. But the gastrin was released from the basolateral surface, went into the blood, and acted in a truly endocrine fashion to bind to those receptors. The other character, I put in blue, I think, was said to be a histamine receptor. The histamine receptor is a particular kind of histamine receptor. It was called an H2 receptor. The histamine that binds to this receptor originated in the enterochromaffin. Like cells. And the enterochromaffin-like cells released the histamine, and in a paracrine fashion, because remember, those ECL cells were neighbors of the parietal cells among the epithelial cells in the gastric pits. And that histamine then would bind to this receptor, the G protein coupled receptor. And then finally, the third one we closed with was another G protein coupled receptor known as a muscarinic acetylcholine receptor. And the question then immediately arose, where does this acetylcholine come from? Well, these cells, these parietal cells, are innervated. These cells are innervated by post-ganglionic parasympathetic neurons. No surprise, the post-ganglionic parasympathetic neurons release acetylcholine. When these post-ganglionic parasympathetic neurons, which of course are part of the rest and digest system, very similar to the post-ganglionic parasympathetic neurons that release acetylcholine onto the acinar cells in the salivary glands, when they bind to this receptor, they start a cascade. All three of these G protein coupled receptors have a cascade which now impinges onto the hydrogen ion potassium ATPase. So in the end, all three of these receptors are running to the same endpoint to increase the hydrogen ion concentration in the lumen of the stomach. We mentioned yesterday, and you may remember, we talked about this acid reflux disease, the GERD, and what that is the incompetence of the esophageal sphincter. And when it doesn't close tightly, then contents of the stomach can move back up into the esophagus. And fundamentally, the way we treat that is with antacids. And there are two major antacids that we'll typically use. The older one is cimetidine. And the cimetidine is an H2 receptor blocker. It's a specific antihistamine binding to the H2 receptor. So it will bind to these receptors and prevent the binding of the normal histamine. So it blocks the effects of histamine. So if you block the effects of histamine released by the ACL cells, you're blocking one of the pathways to increase hydrogen ion concentration, so that reduces the secretion of hydrogen ion into the lumen of the stomach. But the more modern of the antacids, omeprazole, blocks the pump irreversibly. I'm not sure that's right. It blocks the pump irreversibly. So basically, it doesn't work on the basolateral side. It works up here on the apical side. It binds to the pump, takes the pump out permanently. It binds to the active site in the pump, and now takes it and inactivates it. So now one of the selling points of the omeprazole is you only have to take it once every 24 hours. And the reason for that is you've permanently blocked this pump. And the only way you reactivate the release of hydrogen ion is to upregulate genes in the nucleus of these parietal cells to increase the production of protein and put the hydrogen ion potassium ATPases into that membrane. So that's why these omeprazoles really are more effective antacid. Yes?
Could you argue that would more likely increase your chances of cancer if you're having to regenerate those all the time?
So the question here is, are there side effects to the omeprazole? Let me take it in a more general term. And it used to be that we thought these things were benign. Now they're beginning to find it's not quite so simple. That one of the issues that's happening is that when you are long-term blocking the release of hydrogen ion into the lumen of the stomach, there seems to be a lowered uptake of calcium. So it's correlated with osteoporosis in some cases. There's also a situation where we haven't talked about this, but the other thing these parietal cells do is they release a molecule whose name I'm not going to come up with, but it's important for making vitamin B12. And what we'll find too is sometimes people will start to get these anemias because they've blocked the effect of the parietal cells, because they have to acidify the stomach in order to activate the molecule I can't think of that goes to down into the large intestine to activate the generation of vitamin B. So it's probably not so benign as we think. And again, if you're in a world where you're taking in some bacteria from the outside world, then that's dangerous as well, because this hydrogen ion has evolved over hundreds of thousands of years to prevent bacterial growth. So for most of us, it's not a problem, but long term, they're beginning to worry about it a little bit. And you had a question.
Yeah, if that H2 receptor is blocked, is the cell going to accumulate hydrogen ion inside of it?
So the question is, if we block the H2 receptor, will we collect hydrogen ions? Well, that could be true for this as well. But my understanding is no, that once you start getting the hydrogen ion concentration in the cytoplasm up a little bit, then you'll turn off these G protein coupled receptors. There's got to be a feedback system. You won't acidify the cytoplasm. So while this is the homeostatic mechanism here is to make sure you get rid of hydrogen ion and maintain a constant pH in the cytoplasm. And if you don't, if that starts to lower, I'm pretty sure these are going to be closed off. I don't know how that happens, whether it's a pH feedback effect or what, but you don't suddenly acidify the contents of the cell, because you'd kill the cell. You get the pH of the cytoplasm down below 7.2, and it's in serious trouble. Anything else? Yes?
If we're wanting to release hydrogen ions to create a more acidic environment, why then are we going to release the HCO3 as a buffer? I might be getting ahead of us.
You're getting ahead of us, but let me take this question, and then I'll come back to you.
Since it's in equilibrium with, what is it, like the pulmonary capillary or something, wouldn't it just start reversing that reaction? Because didn't you say like a couple lectures ago that the H plus and the HCO3 are like interchanging through that capillary?
So a fair point here too, is that if we're dealing with the carbonic anhydrase, if we get levels in here of bicarbonate high enough, then we're going to run this reaction in the opposite direction. So you're right, then we'll get CO2. So that may be some of the explanation as well. And for your question, let's we can begin that in a moment. But what we're going to see is we're going to squirt that acid into the small intestine, and we're going to use the bicarbonate to absorb that hydrogen ion and go back to CO2 and water. Right. And we'll come to that. So anything else? Let's take a minute then, though, and talk about the anatomy of the small intestine. We'll begin over here with the idea of the tunics. And wherever you look along the length of the small intestine, even the large intestine, you find the same organization of tunics. And this will be reminiscent of what we talked about yesterday in the stomach. So the mucosa itself is thrown into these folds. And this here is meant to be the lumen. This is meant to be the mucosa. Immediately beneath the mucosa will be the submucosa. And beyond the submucosa, just as we saw yesterday, attached to the submucosa will be a layer of longitudinal smooth muscle. And then around that will be a layer of circular smooth muscle. And then finally, attached to the outer layer of smooth muscle will be a continuous layer of mesothelial cells. So this is the mesothelium. Sometimes known as the serosal surface. But the pattern is the same. You go from a mesothelium to smooth muscle to submucosa to mucosa to the lumen of the tube. I need to spend a moment here describing the regions of the small intestine. Now, if you take an anatomy course, you'll spend a lot of time talking about these. But here we're just going to lay out the three major regions and pretty much leave them behind. The first is the duodenum. The second is the jejunum. And the third is the ileum. Now, the duodenum itself is a pretty short region. It's only, in the neighborhood of about 12 inches long. And in fact, there's an easy way to remember this. The duodenum comes from the Latin duodecum. Duodecum means 12. And the story is one of the early anatomists, probably in the late Middle Ages, early Renaissance, when they first started doing gross anatomy, found the section of the small intestine, recognized there was a distinction between this initial segment of the small intestine and the next segment. And he did a crude measurement, basically took his finger and walked his finger along the duodenum. Turned out there were 12 finger widths. 12 finger widths is 12 inches. That's a fat-fingered anatomist. I'm not sure that Donald Trump imagines his hands are that big. But you can remember that the duodenum is named from duodecum. Jejunum also comes from the Latin meaning empty. Because again, in the late Middle Ages, early Renaissance, when they first were doing these dissections, they were doing them on people who'd been executed. These people hadn't eaten for a while. And so there was nothing in this region of the intestine, so it comes from the Latin jejunum, meaning empty. And we still use this word sometimes. When we want to insult somebody, we say they're jejune, They're empty or without character. And so this again comes from the Latin. Finally, the ilium, I think, is just related to the iliac crest, which is part of the pelvis. But these are the major regions. This is the smallest of the regions. Most of the absorption occurs across the mucosa of these two regions, the duodenum and the jejunum. But if we begin thinking about what's going on in the duodenum, we basically take our crude drawing that we had yesterday. Well, up here we describe this as the esophageal sphincter. And down here, the pyloric sphincter. We described yesterday how the esophageal sphincter was a circular piece of smooth muscle and regulated movement from the esophagus into the stomach proper. At this end of the stomach, the pyloric sphincter regulates movement of chyme into the small intestine. So now this is the small intestine here. We can label this. Let me make it a little bigger so I can work with it. This will be the duodenum. The mucosa will be composed of epithelial cells known as enterocytes. So these are the enterocytes. These are the enterocytes of the intestinal mucosa. Now remember, the contents of the stomach is chyme. And this can have a pH of less than 2. That's pretty acidic. So if the signal comes from the brainstem to open the pyloric sphincter, then the contraction of the smooth muscle surrounding the stomach will shoot about 30 to 50 mils of the chyme into the small intestine. The epithelial cells of the small intestine are for absorption. They're basically these epithelial cells across which we're going to do epithelial transport. These cells lack the overlying mucus layer that we saw in the epithelial cells, the mucosa of the stomach. So these cells are susceptible to damage by low pH. They're susceptible to damage by the proteolytic enzymes. So we have to modify the contents coming in from the stomach pretty rapidly. And that's where we're headed now. There's a structure here. I'll put it in purple. I'm going to exaggerate its size so I can work with it a little bit. This structure is meant to represent the pancreas. The pancreas has a tube that enters directly into the small intestine. Now, if you get down to the nuts and bolts of this, you're going to find that there's a tube that comes from the gallbladder, the common bile duct, and the pancreatic duct fuse to make a common bile duct that goes into the small intestine. Don't worry about that. All I want you to recognize is that we have a tube that connects the lumen of the pancreas with the lumen of the small intestine. And if we follow the structure of the pancreas back here, we see that it's thrown into these loops. But it's composed of an epithelium. So this is meant to be the epithelium of the exocrine pancreas. It's an exocrine gland, so if we were to stand on this epithelium, we could walk on this epithelium and walk on a continuous epithelium through the duct, join the epithelium in the mucosa of the small intestine, and go to the outside world. So it's a proper epithelium. The epithelial cells of the pancreas are broken into two regions. These are the so-called duct cells. And there are these bulbous structures here, which are composed of epithelial cells called acinar cells. So the players here are going to be, first, I'll put this in orange. There's a cell down here, a cell type in the mucosa of the small intestine in orange, which is known as an S cell. And then there's going to be this brown cell here. This is called an I cell. The S cells and I cells are modified enterocytes in the small intestine. So let's go back up to the upper panel. And what we're going to see here is that the S cells are a kind of pH monitor. The S cells are using their apical surfaces to monitor for the hydrogen ion concentration, the pH, in the lumen of the small intestine. So it's no surprise that when the pyloric sphincter opens and a squirt of chyme comes into the small intestine, that the contents of the small intestine, at least briefly, are going to have the pH plummet. So when the chyme comes in, what the S cells do, I'll draw this as an S cell. It's meant to be an S cell. It sits on a basal lamina. It's a mucosal cell. It's an epithelial cell, so it has a basilar region. And it has an apical region. The apical region monitors pH. And hydrogen ion will signal the cell to release or secrete the hormone secretin. Secretin is a peptide hormone. Over here, I'm going to put in blue, this is going to be a pancreatic duct cell. Again, it's an epithelial cell, so it sits on a basal lamina. And it has a corresponding apical surface. Here on this pancreatic duct cell, on the basolateral side, is a G protein coupled receptor. This is a GPCR that binds secretin. So again, these apical membranes of the S cells see low pH. That signals the release of a hormone, secretin, it's a peptide hormone. It travels through the blood. It travels through the blood in a typically endocrine fashion, comes over here, and then binds to this G protein coupled receptor on the basolateral side. And what this G protein coupled receptor does is it upregulates carbonic anhydrase. Now remember, the basolateral surface here is going to be not far from a capillary. And so carbon dioxide in this capillary can leave the blood vascular compartment and get into the cell. But remember what will happen with carbon dioxide plus water in the presence of carbonic anhydrase. It's going to convert the carbon dioxide and water into a hydrogen ion and a bicarbonate ion. This is exactly what a parietal cell does. But remember, the parietal cell in the stomach is going to move that hydrogen ion out across its apical surface and deposit that in the lumen of the stomach. We're going to turn this one upside down. What we're going to do is we're going to take the hydrogen ion, put it back into the blood vascular compartment, but then out of its apical surface, we're going to secrete bicarbonate, probably through a chloride bicarbonate exchanger. But be that as it may, what that means then is we're pumping bicarbonate, we're moving bicarbonate into the lumen of the pancreas. That bicarbonate now is free to move through the pancreatic duct into the common bile duct and down into the lumen of the small intestine. The bicarbonate now can act as a buffer. And we can run this thing in the other direction. When we have HCO3 negative plus hydrogen ion, we can now run this back to CO2 plus water. So now we're buffering the hydrogen ion, we're converting it into water and CO2, and we do that in the small intestine. That now will bring the pH from in the range of 2, which is the pH of the chyme, into the range of about 7.2 to 7.4. We can do this because we have an enormous amount of bicarbonate. If you look at the pKs of bicarbonate, they're terrible for this. I think they're up around 8 or 9, somebody said. But in fact, we can do this because we have such an enormous amount of bicarbonate. The long and the short of it is the bicarbonate is a buffer. So we've now used the hydrogen ion coming into the small intestine, the chyme, to signal the pancreas to release bicarbonate to now buffer the hydrogen ion. But there's another character in here that we described, which is the eye cell. Let's go back here. This brown cell, which is the eye cell, this is the eye cell. It too is oriented. It's an epithelial cell. It's a modified enterocyte. It sits on a basal lamina. Now in this case, the apical surface here recognizes lipid and protein. It recognizes lipid and protein in chyme. By recognizing the lipid and protein in the chyme, it then again mobilizes vesicles. And the vesicles now release a hormone. They secrete the exocytosis, the hormone cholecystokinin. We abbreviate cholecystokinin as CCK. But if it helps you remember it, there's actually some meaning in this word. Chola means bile. Cysto means sac. And kinin means move. Bile, sac, move. Before we understood the effects of CCK coming from the eye cells on the pancreas, it was known that cholecystokinin, if bound to smooth muscle cells in the gallbladder, would cause smooth muscle contraction of the gallbladder and squirt bile into the small intestine. So in response to fat, it would squirt in some bile. But now it's an extension of that. These brown eye cells are recognizing protein and lipid, releasing cholecystokinin. The cholecystokinin then finds its way to the epithelial cells here known as the acinar cells. Again, it's a classic G protein coupled receptor. So there's a G protein coupled receptor on the basolateral side. The CCK binds to it, and the CCK binding to this cell signals the release from the apical surface through vesicles, digestive enzymes. These digestive enzymes include pancreatic amylase. They include lipase. And they include trypsinogen. Here's that suffix again, OGEN, it's inactive. Trypsinogen and a related molecule called chymotrypsinogen. The pancreatic amylase is going to work in the small intestine to convert starch into maltose. The lipase, we're going to see, is going to work in the small intestine to convert lipid to monoglyceride. And fatty acid. The trypsinogen and chymotrypsinogen will become activated proteases that convert protein to peptide. So that's the list of players. But the signaling here is such that when the chyme comes into the small intestine, it tells the S cells to release secretin. The secretin binds to duct cells in the exocrine pancreas. When the secretin binds to the duct cells, we release bicarbonate to offset or neutralize the acid. Similarly, when the contents of the stomach come in that are rich in lipid, rich in fat, that signals the eye cells to release the cholecystokinin. The cholecystokinin, again, is a hormone, acts in a classically endocrine fashion, comes over and binds to epithelial cells in particular the acinar cells in the pancreas. That signals these cells to release the digestive enzymes, which again follow this tube down into the small intestine. Yes?
Small question. What kind of hormone is the CCK?
CCK is peptide hormone. It's a classic peptide hormone. Truth is, it looks an awful lot like gastrin. Any other questions? Well, in the last few minutes then, let me take this back panel and what I'm going to do is I'm going to take a section out of here, out of this intestinal mucosa, and I'm going to blow it up. And what I want to draw on this back board is a villus. And I'll use some of our shorthand to try to describe it in the sense that there will be a single layer of epithelial cells. And these vertical hashes here, now horizontal, are meant to represent the division of this epithelium into individual enterocytes. We can say these are the enterocytes of the intestinal mucosa. These are classic enterocytes in that they have an apical surface. The apical surface now faces the lumen of the intestine. So if we were to go up to this upper panel again, the epithelia here, epithelial cells, will be facing the lumen of the tube. But the epithelial cells are folded into these finger-like structures called villi. So you have a continuous layer of epithelial cells. Again, they always sit on a basal lamina. They are, after all, epithelial cells. That gives us an orientation where the apical surfaces are facing the lumen of the small intestine, and the basilar surfaces are facing this extracellular space. But this extracellular space is occupied. It's occupied by an extension of a blood vessel. See if I can branch it. So this is meant to be an extension as a capillary of the hepatic portal vein. So all of the blood coming into the gut is coming through this portal system. And the blood comes through and it will flow through these capillaries. Now, these are classic capillaries, and so these cells, if we blow this up a little bit here, these epithelial cells sitting on their basal lamina are right next door to the endothelial cells forming the capillary of the hepatic portal vein. So the blood flow through here is going to be that the blood is going to flow in through these tubes, and the endothelial cells are now strategically located so that the movement of glucose and amino acids, which are the final breakdown product of starch, disaccharides, and proteins, these endothelial cells are strategically located so that now these water-soluble molecules can move across the epithelium into the extracellular space and now into the blood vascular compartment of the portal vein. The blood here in the portal vein is destined for the liver. So hold on. Do I have enough? Let me show the last couple minutes here. Let me use this to show this slide because I think it's a slide that summarizes this pretty well. So this is a pretty picture, a really nicely drawn image of a villus. And it's worth taking a moment to look at this because this is a multicellular structure. This is a continuous layer of enterocytes. The apical surface of the enterocytes faces the lumen of the tube. The basilar surface faces the other side, and it's right adjacent to these capillaries that are branches of the hepatic portal vein. So water-soluble nutrients can come across the epithelium of the small intestine and enter the blood vascular compartment of the hepatic portal vein, and those nutrients then will be delivered to the liver where they're going to be reassembled. The other character in here you'll notice in green is actually a lacteal. That's actually a capillary of a lymphatic vessel. It turns out this will be responsible for carrying lipids. We're going to see on Monday that the lipid function here is we're actually going to release lipids in a form called chylomicrons that are too big to get into the blood vascular compartment. And instead, because the gaps between adjacent endothelial cells of the lymphatic vessel are bigger, they'll pass right into the lymphatic system and then be dumped into the subclavian vein. And that's one of the reasons why after a big meal, if you look at plasma in blood, it's cloudy because it contains all those lipids. And we'll bring that idea up on Monday.